Ever watched a crime show and seen them talk about DNA evidence? It sounds like something straight out of science fiction, but it's a real, powerful tool used to solve crimes, prove paternity, and even identify ancient remains. Today, we are pulling back the curtain on this incredible technology and showing you the magic behind the science, the 6 fascinating steps of DNA fingerprinting. Think of your DNA as a massive instruction manual. It's what makes you, well, you. But tucked within that manual are special sections, unique to each of us. We are talking about repeated sequences of DNA, like a genetic stutter. These are our variable number tandem repeats, or VNTRs, and they're the key to creating our unique DNA fingerprint. So, how do scientists turn this microscopic information into a visual pattern? Let's break it down, step by step. First things first, you need a sample. This can be a drop of blood, a few cells from a hair follicle, or even a swab of saliva. Basically, any biological material that contains cells with a nucleus. The goal here is simple: get the DNA out. Scientists use special chemicals to break open the cells, separate the DNA from all the other cellular junk, and purify it. It's like finding a single, precious thread in a giant pile of laundry. Now for the fun part. We have a long, continuous strand of DNA, but we need to chop it up into manageable pieces. Enter our super-specific genetic scissors, restriction enzymes. These are proteins that act like highly trained security guards, recognizing and cutting the DNA only at specific, predetermined sequences. Because everyone's VNTRs are different lengths, these cuts create a unique collection of fragments, like a personalized jigsaw puzzle. We have our chopped up DNA. Now we need to sort it. This is where electrophoresis comes in. Think of it as a genetic obstacle course. We load our DNA fragments into a special gel, which is full of tiny pores. We then apply an electric current. Since DNA has a negative charge, it's pulled towards the positive end. The smaller, lighter fragments zoom through the gel's pores easily, traveling farther. The bigger, heavier fragments get stuck and move much more slowly. The result? Our DNA is neatly separated by size, creating a unique ladder of bands. The gel is great for separation, but it's a bit fragile. To make our DNA fingerprint permanent and easier to work with, we need to transfer it. This process, called blotting, involves carefully pressing a sheet of nylon or nitrocellulose membrane onto the gel. This effectively lifts the DNA fragments right off the gel and onto a much more stable surface. We have a membrane with our unique DNA pattern, but it's invisible to the naked eye. We need to make it stand out. This is where labeled probes come in. These are small, synthetic pieces of DNA, designed to be the perfect match for those VNTR sequences we talked about earlier. We also tag them with something special, either a radioactive marker or a fluorescent dye. When we wash these probes over our membrane, they seek out and stick only to their complementary DNA sequences. It's like sending out a searchlight to highlight the exact parts of the DNA we are interested in. Autoradiography, or imaging. The moment of truth. After the probes have done their work, we take our membrane and place it against a special piece of film, an X-ray film if we used radioactive probes, or we use a special scanner for fluorescent ones. The energy from the probes exposes the film, and when we develop it, we are left with a series of dark bands. Each band corresponds to a DNA fragment of a specific size. And because everyone's VNTRs are unique, the pattern of these bands is a one-of-a-kind DNA fingerprint. And there you have it. From a single cell to a distinct, scannable pattern. This powerful process allows scientists to create a unique identifier for any individual on the planet. It's a testament to the incredible power of biology and a true game-changer in forensic science. I hope this gave you a new appreciation for the science behind DNA evidence. If you found this interesting, be sure to like this video, subscribe for more amazing science breakdowns, and let us know in the comments what other scientific processes you'd like us to demystify. Thanks for watching.